Hi, my name is Nitin Vincent. I'm a third-year PhD student in the Biological Sciences Department, advised by Dr. Pfrender and Dr. Jason Rohr. I'd like to talk to you about my research today. So I'm actually involved in a couple of different water-related projects. My thesis work revolves around a molecule called tetrodotoxin, which is produced by a lot of different aquatic organisms, including pufferfish, blue-ringed octopus, and salamanders. I'm looking at caddisflies, which are aquatic insects that are resistant to this toxin, are able to consume the eggs of highly toxic salamanders on the west coast. The toxin works by blocking sodium channels in its victims, which is also how the most common insecticides used today work. So I'm looking at the genetic mutations responsible for tetrodotoxin resistance in caddisflies and analyzing them to see if they also result in resistance to insecticides, which could be a problem for aquatic ecosystems. The other project I'm currently working on is a zooplankton biodiversity assay in the Great Lakes. Our collaborators collect zooplankton samples from around 100 sites at various locations in the Great Lakes system, send them to our lab here. I extract the DNA and search these samples for DNA fragments called barcodes, which tell us what species of zooplankton are present in each sample and in what ratios. This allows us to create a geographical map of zooplankton species diversity in the Great Lakes. This can potentially be used to look for indicator species, which help in biomonitoring. Essentially, the presence or absence of certain species in an area could be an indicator of certain phenomena, like differences in temperature, high pollution or microplastics abundance, fish stock declines, or presence of invasive species. Biomonitoring of zooplankton populations can help us intervene early and stop these problems from getting out of hand and preserve the ecology of the Great Lakes. So I was working on two similar projects at my undergrad institution that translated into water-related work when I came to Notre Dame. With tetrodotoxin, I was actually looking at the production of tetrodotoxin in newts rather than resistance in caddisflies. I was also looking at bacteria community compositions to find indicator species for heavy metal pollution. At Notre Dame, because we're close to the Great Lakes, we have collaborators who exclusively do water-related research. And I became involved with them thanks to my work on similar problems previously. Tetrodotoxin production became tetrodotoxin and aquatic insecticide resistance, and bacteria indicator species became zooplankton indicator species. I think the applications of my work to society makes me the most excited. It would be great if down the line my work could help in designing more effective insecticides that are ethical and better for aquatic communities, or if a zooplankton biomonitoring survey is set up to warn authorities about potential changes in lake ecology so they can intervene. Also, I just really like the exercise of fiddling around with DNA and using bioinformatics to solve these problems. As I mentioned earlier, none of this work would be possible without our collaborators. We are primarily a genomics and evolutionary biology lab, so we have expertise in DNA sequencing and analysis. But in order for our work to have the highest impact, we need to collaborate with groups who have other necessary knowledge, like pollution data, or sampling procedures, or setting up biomonitoring programs, or science policy initiatives. Water has such far-reaching impacts on everyone, so scientists in every field are involved in water research. And I think harnessing all of this knowledge leads to the most impactful research. Thank you.